Okay, today I want to show you how to do your sequence identification to identify your isolate. So unfortunately, because of the shutdown, we were not able to sequence your specific isolates. So we're going to be giving you sequences from last semester. And then you'll build your report for the final report in the lab based on the information that you learn about this particular isolate and the information you generated prior to us not being in lab anymore. So what you'll receive from your TAs are files. They'll be group 1, group 2, whatever, and you'll receive one that says F and one that says R, and then they'll have this ending .seq. These files can be opened with a text editor. So if you're on a Mac, I use TextEdit. If you're on a PC, I believe the program is called Notepad. You may have to force it to open that way. And so I'll show you kind of how you can do that. You could go again on the Mac. I'm going to open the test F, open with TextEdit. So you can force it to open with the program you want. Okay, so what we have to do, there are a few different steps before we can identify our isolate. What we do in this sequencing procedure is you guys PCR amplified your 16S rRNA gene, and you generated a fragment that was approximately 1,300 base pairs. So if we were able to continue this semester, you would have next run your PCR products on the gel, and I showed you one of the gels in the video from Monday, April 13th, and you could see how bands would look on a gel. The step after that is what's called PCR cleanup. We basically remove all of the primers, we remove all of the other chemicals that allow us to do PCR, and we have a purified DNA product. Then what we do is we take that purified DNA product and we split it into two tubes. In one tube, we add what's called the forward primer. It will bind to one strand of DNA and allow amplification in the 5' to 3' direction. In the other tube, we will add what is called the reverse primer. The reverse primer will bind to the other strand of DNA and will amplify in the 5' to 3' direction. What I'll add at the end of this video is a little brief tutorial about how PCR works, and so what we would actually be seeing. So you'll need to understand how PCR works. We then would send those tubes, one containing the forward primer, one containing the reverse primer, to a company called Eurofins, and they do what is called Sanger sequencing. We talked about Sanger sequencing in lecture, so I will expect that if on your lab practical you are asked how Sanger sequencing works, you should be able to explain it. It is also known as dideoxy sequencing, so it is important that you understand how that works. We then get 2 files per sample that we send. The first file is the forward reads, and you'll see that there are some ends, and then we have the 4 different bases. The ends represent an area where the laser could not determine which base was in that position. We often see ends near the beginning and near the end of a sequencing read as the polymerase is trying to get going and as the polymerase kind of peters out. So it's a different type of polymerase that we use for doing sequencing. So this is our forward read. We will also have a set generated from the reverse read, so reading from the opposite strand in the other direction. So we have our forward and our reverse. So the first thing that we have to do is make it so that we can align these two sequences together and get what's called the consensus sequence. We want to be able to put our forward and reverse together in such a way that we can predict what our sequence actually was. And so because we read in opposite directions, forward and reverse, we have to be able to turn our reverse reads into the appropriate orientation, and it's called a reverse complement. So basically, we want to turn the bottom strand of the DNA, flip it around, and turn it into the top strand so that we can then align our two strands and get the consensus of what our sequencing results were. So in order to do that, you should navigate to this website here, bioinformatics.org/sms/rev_comp.html. We take only our reverse sequence, copy, and paste, and submit. Okay, so it has done the reverse complement, and here's how you can tell. My sequence ended with AA, so my new sequence starts with TT. So I complemented, and I'm reading it backwards. So we'll select this, copy, and then we'll navigate to another website. This website is called Emboss. This is what allows us to merge our two reads to get the consensus sequence. So this is bioinformatics.nl/cgi-bin/emboss/merger. You could also Google emboss merger. So I'll put the reverse read, the reverse complement reverse read down here in the second box. It doesn't really matter, but that's what I'm going to do for clarity. I'm going to pull up my forward read, copy, paste, and then this part's really easy. Click, run merger. That didn't take very long at all, maybe just a couple of seconds. And now you can see where the sequences kind of started to line up. And there's some areas where maybe they didn't line up perfectly. And again, this could be because of errors in the polymerase and errors in the sequencing. And as we start to get kind of towards the middle, we see much more clarity in sequencing, and we have a lot of consensus. This is really what we're looking for. This is why we have to line these two sequences up. And so then again, you see at the end, it didn't really line up very well. And so it tells you places where there were errors, but we're going to scroll all the way down to the bottom. And down here we now have our output file where it lined it up the best it could. And we will copy that as well. So I might, if I were you, save this in another document so you could open up another text edit, or you could open up a Word document. It doesn't really matter. So if I open up a new text edit right there, and then I can call it, oops, I don't want it to be so big, but that's okay. I'll call it consensus. Okay, shrink it back down. All right, so then I'm gonna make sure that I have selected and copied that sequence. And the final website we'll go to is BLAST, Basic Local Alignment Search Tool. You can see the address up here, blast.ncbi.nlm.nih.gov/blast.cgi. You could also Google NCBI BLAST and you'll be able to find this website. We are going to do what is called a nucleotide blast. We have a DNA sequence and we are going to search the entire database of DNA sequences that is held at the National Institutes of Health to try to figure out what species our DNA came from. So I'm going to paste my sequence in there. You can leave all of the standard defaults, and then we will go ahead and click BLAST. This might take a couple of minutes. The page will continually refresh itself. If it ends up taking a really long time, I'll of course cut the video and then bring it back up when the sequencing results are done. All right, so my sequencing is done. And so it gives you a page that looks like this. And there's some important information that you can get from this website. And this is going to be information that you'll need to include in your final report. It will also be information that you'll need to include in your lab report for Wednesday, April 15th. I want to know what my species is, and so what I have now is a list of the top hits, what my species matched found in the NCBI database. And looking at all of these hits, these are the top 100 hits, it is pretty clear that my isolate is a member of the genus Bacillum, and it's most likely Bacillus megaterum. So I'm going to look at my top hit. You can see it matches the 16S ribosomal RNA gene. So we click on our top hit, and here you can see how our sequence, the query sequence, matched the subject sequence, which ended up being Bacillus. So you see there are lots and lots of identical bases. This tells you right here, identities, how many bases are the same out of total number of bases where there is an alignment. So 1317 were the same out of 1319. And you can see right here in our query sequence, there was an N that didn't match a C found in the subject. And here's an N that didn't match a T. Those were the only ones that didn't match. So I can be fairly confident that my strain, my isolate, is probably Bacillus megaterum. Here is a value called the expect value, or the E value. This number basically represents the percent of the time, or the probability that you would expect this result by chance. And you can see that my expect value is 0. So this is not a result that was generated by chance. This is the true result. You can look at a graphic summary that shows you the alignment. You can click on your top hit and it'll tell you, oh, Bacillus megaterum. You can go back to taxonomy. It shows you where Bacillus megaterum kind of fits in here. And you can go back to the descriptions and see again how good your match was. So you want to make sure that you identify which match is the best, because you need to know which organism you have and how to justify which organism you have for your final report, and also for your laboratory report due on April 15th.